All right. Good morning, part two for Friday, April 10th. Just posted a chapter 24 concept check conversation, and now I'm going to jump into a chapter 5 reading guide discussion. Before we do, I'm playing a little Bill Withers this morning. He passed away this week, and just a legendary artist. And so this is one you guys probably know a little bit better. So let's get to where we need to get to. As I said on the other video for today, guys, your quiz on Monday covers chapter 24, sections 1 and 2, and chapter 25, sections 1 and 2. That's it. There is no chapter 26, as I had mentioned earlier this week. There might be. To get through this week, I kept it simple for y'all, and that will be what you'll see on Monday is that quiz over those 4 total sections. The 3 things that you'll want to be looking at to study are the video lecture of the PowerPoint that I gave on Monday over chapter 24, sections 1 and 2, which is really covering types of reproductive isolation, species concepts, and allopatric and sympatric speciation. The concept check questions that I gave you over chapter 24, and then finally this, which is our reading guide for chapter 25, which is a quick overview of the entire history of the origin of life on Earth. So let's take a look, and I'll let you know what we need to focus on. All right. Again, I highly suggest you find a playlist for Bill Withers and just let it play while you take care of some work today and tomorrow. All right. Let's have at it, folks. History of life on Earth. Yeah, that's a pretty big conversation, pretty big topic that I'm not about to cover in 20 minutes. So we are giving an overview of things to at least give us an idea of what we know and understand from our research as far as what potentially could have happened over 3.5 to 4.5 billion years ago that started life here on Earth. As with any of these conversations, evolutionarily speaking, as we go back in time and rewind, this is based on research via fossils that we gather, and we radiometrically date them, and I'll talk about that here in a little bit. And also trying to recreate situations that you'll see here in a few questions, recreate early Earth situations in the lab to see if we can potentially see these things that may have spurred the origin of cellular life 3.5 to 4 billion years ago. And so when we look at this process, as we look at our chapter 25 reading guide, that's called macroevolution. Remember, microevolution really was chapter 23, where we were looking at a specific population, and we were monitoring that population from generation to generation to really just pinpoint allele frequencies changing. And again, that was complicated. That was tough stuff to think about and conceptualize and mathematically understand. Here we take a step back. We zoom out, and we're looking at this big, broad process of how could we get to, through this lens of natural selection and through this lens of life adapting and changing to environmental situations and pressures here on Earth, how can we get to what we have here today on Earth, which is such a mass diversification of life, from singular life, single-celled life, like bacteria and archaea and protists, to the incredible multicellular life that we see here on Earth. That's macroevolution. What we have currently, the oldest fossils of life on Earth, actually are incredibly old. We have fossil evidence of single-celled unicellular life of 3.5 billion years old. And that's come as techniques have sharpened and gotten better as technology has improved. That 3.5 billion years has not changed much. As again, I learned about biology originally, for the first time last century, and we were still taught that number of 3.5 billion years. So that really hasn't changed, even as technology has vastly improved in my 40 plus years. That number really hasn't changed much. And what we want to look at number 3 is the hypothesis of the 4 stages, kind of the step-by-step of, based on what we know and understand, what are we looking at potentially occurred to lead towards cellular life. Well, we have to start with molecular first. Before there were even cells, there were molecules here on life. And most of them were just inorganic molecules. And what we had early on, and as far as our understanding and thinking goes, are the building blocks. The original synthesis of things like amino acids. And remember, amino acids, we put those together, we're building proteins. Nitrogenous bases, the synthesis of those and the beginnings of those molecules then led to those monomers getting put together to build things like RNA and DNA. So that's our step B here. Our second stage is those monomers actually anabolically synthesizing and getting put together to build those larger macromolecules, those macromolecules of life, proteins and nucleic acids. And then finally, as we go past that, sorry, not finally, but our next step, our stage 3 is having those molecules able to survive. And to be able to survive means the harsh environments of the molecular world meant protection. And we note that protection comes with, in our understanding today, is of cellular membranes. Cell membranes give that protection. And so before cells came to be, there's this precursor, or this before there were cells, they're called protocells. And they really weren't as advanced as the full-blown lipid bilayer with all the other things that we know to come with a cell membrane when we think about that fluid mosaic model that we learned about in chapter 7. But these were the basic beginnings of a lipid bilayer. And so we had droplets that were actually surrounded by a membrane, a lipid bilayer of sorts. So there's your protection. But if we just have one protocell like that, that's got, these amazing proteins and these amazing nucleic acids, and it survives and it floats around in its world, woohoo, survival for that one protocell. But what does that mean for evolution of life? Not much until stage 4. Stage 4 is when some of those molecules within that protected protocell are able to replicate their information. And that's when we start to think about genetics and heredity. We start to think about genetics and heredity, it's the genetics of one generation being able to copy itself and pass itself on to the next generation. Think simplistically like mitosis, copying of DNA, split that cell. If those things started to happen, or when those things started to happen, now we're moving in a direction of actual cellular life surviving, not just for one generation, but for generation after generation after generation after generation, and so on. So as we go down to number 4, our full-blown timeline, again, a number that has not budged in my lifetime, is our knowledge of the beginning of Earth. Doesn't mean that that's hardcore fact. Scientific evidence continues to update and change, but more evidence has supported that Earth was formed 4.6 billion years ago, basically around the time of our entire solar system. And if you really want to go even further back, our evidence of the formation of the universe that's actually a number that has changed considerably in my lifetime. The understanding of the origin of the universe is anywhere from about 11 to 13 billion years ago. Just some numbers in terms of the universe. But for our planet, our moon, our solar system, our sun, we're looking at about 4.6 billion years ago. Early on, there was not a lot of oxygen on planet Earth. Like, we would not have been able, as humans, oxygen-breathing, aerobic, respiring organisms, we would not have been able to survive because there was very low oxygen. And there was lots of water vapor. And then approximately 4 billion years ago, there was kind of this absence of water as we kind of do this fast forward through our macroevolutionary look of things. There was so much stuff going on in the early solar system, so much rock and ice that was just bombarding not just planet Earth, but all the planets of the solar system. And through all those crazy collisions that were happening, there was such heat that was generated here on planet Earth that the water vaporized. And so we really didn't have much water for that time period. And again, I'm kind of picking and choosing a few key points. This isn't going to fill in all the gaps of your understanding. If this truthfully interests you and you want to know more, you have questions about, like, wait, how did Dupree jump from step 5 to step 6? Again, this not being a full-blown class on earth science and understanding the beginning of the earth, I'm kind of picking some things to help us at least get a progression of understanding. As we look at question 7, scientist in the 1920s, J.B.S. Haldane, he hypothesized that the energy needed to stimulate the energy needed to build these molecules, thinking back to our early stages of synthesis of amino acids. Remember, to synthesize things, there has to be energy to anabolically build molecules. So where's that energy come from? Well, lots of lightning storms and loads of UV radiation. Early Earth, due to lack of oxygen, really didn't have an ozone layer. So there wasn't that protection of ozone to shield us from the amount of protection we get nowadays from the sun's UV radiation. And so back then, the Earth was really just getting hammered with UV radiation. And so his hypothesis was that was an energy source along with these lightning storms. Those energy sources could have fueled biosynthesis, or the anabolic building of these monomers, our amino acids, and our nucleotides, which then could have led to the biosynthesis of putting those together to build the big things, to build the big macromolecules. So Miller and Urey, we're talking about two scientists. We're fast forwarding about 30 plus years. Miller and Urey, they were able to take this hypothesis of all these things that were in the beginnings of planet Earth, a large called primordial soup. And primordial just means it was there at the beginning, a primordial soup, if you will, of inorganic molecules. And then they provided energy through electrostatic sparks. And through that kind of cool in a lab experiment of what Earth could have been like, some organic molecules, some amino acids, and beginnings of RNA molecules were created. And since then, some scientists have been able to recreate this experiment. As much as that wasn't everything that we'd like to see, we all of a sudden didn't get this little biosphere that created the beginning of bacterial life, because this occurred over so many millions and even billions of years. That's not something that could have been really recreated, but it at least supported a lot of our hypotheses about how things could have been. So as we go down to number 9, organic molecules may have also formed near volcanoes or deep sea hydrothermal vents, where again, there was a lot of energy through the heat that's produced through volcanoes and the heat from a hydrothermal vent. We're going to the very bottom of the ocean. These hydrothermal vents put out a ton of heat that's coming directly from the center of the Earth. Down to number 10, explain how organic matter may have actually come from meteorites also. So we have research from a meteorite that hit Australia in 1969 called the Murchison meteorite. And it actually contained on that meteorite that came from outer space over 80 amino acids. Remember, we have 20 amino acids that make up life here on Earth, but there were over 80 amino acids on that meteorite. So some of them have never even actually been found on Earth elsewhere. And that meteorite also contained some precursors and organic molecules that could have led to life. So that's just more into the mix of what scientists and researchers and geologists are trying to understand about what could have been early life. I talked about protocells earlier. These precellular structures may have just been vesicles. Remember, we've talked about vesicles in our cells. They're just fluid-filled compartments enclosed by a simplistic membrane. It's still got that lipid bilayer without all the bells and whistles of a cell membrane. And let's see. Recent experiments show that they actually can exhibit certain properties, simple properties of life. There's a continued conversation about what was the first genetic material, and more research points to RNA starting things, and then DNA evolved from RNA. Because RNA has been shown the ability to actually catalyze its own replication or make copies of itself, which DNA has never been found that it's been able to do that. So again, if we go back up to our stage 4 of origin of self-replicating molecules, there's our piece right there. RNA does show the capability to copy itself and act like an enzyme. So that's where we see that term ribozyme. And then we get these protocells created that they increase in numbers by the simple approach of Darwin's natural selection. Some survived better and were able to pass on their self-replicating molecules of inheritance. And then RNA provided that template for the evolution of DNA as time went by. DNA has, though, become much more common as far as the inheritance material or molecule of life here on Earth than RNA. And your reasons is that DNA is much more stable chemically. RNA is fragile. It can be broken up much more simply. And that DNA can be replicated more accurately. There are less mistakes, less mutations that allow for the continuance of a species. As we go down here into 25.2, again, our last section of chapter 25, looking at our fossil record, different types of rock that are out there, sedimentary rocks, igneous rocks, etc. Sedimentary rocks, those are the richest source of fossils. So if you're in an area where it's mainly, say, igneous rock that doesn't produce many fossils, then whatever life lived there, we may not see much fossil evidence. So I know we spoke about this earlier in this unit where fossil evidence is kind of a hit or miss in terms of what we're able to find, and we have to make the best sense that we can of what we find. Two other ways that organisms are preserved as fossils other than in rock are insects that can be preserved in amber or the sap of trees, and also mammals preserved in ice. Limitations, again, we have spoke about this. Many organisms just didn't die at the right place at the right time to be preserved as fossils. And also just through geologic processes, the changing of our Earth through tectonic plates moving. You guys spoke about the two plates that are colliding to create gosh, I'm slowing down for words here, but forming mountains like Mount Everest and all the huge mountains that we see in Southeast Asia. Just through process of creation of mountains, tons of fossils get destroyed through that. And of course, we can't dig everywhere, so many of them just haven't been found. As a result, the fossil record is biased in favor of species that existed for a long time, so there's just a lot more of them and they're easier to find. And they had hard shells or skeletons that lent to fossilization. We spoke about this before we went on break. The deeper a fossil is, the older it is. But this just tells us that relative age of that fossil. To find the absolute age, you got to use radiometric dating, which means finding radioactive isotopes in that rock. And the rate of decay of an isotope is measured by its half-life, which we're able to actually pin down based on that radioactive isotope. Uranium, that's radioactive, decays into lead, for instance, at a very specific rate. And so if we're able to take a look at a rock and see that radioactive uranium and compare how much there is of that to how much there is of that lead that it decays into, scientists can determine how old that rock is. And so that's been insanely instrumental in scientists determining the age of rocks, going back to those crazy numbers that I mentioned at the beginning of this 3.5 billion year old fossil. Yeah, it's not a guess, and it's not a scientist just looking up at the sky and saying, boy, I wish the Earth was this old. No, again, this is all related to the evidence that we have, that we find through the technologies that we've developed over the course of time. Finally, just for a connection to fossils, when we are looking at a fossil, a skull of one organism, and we see another skull that looks super duper similar, scientists have been able to gain enough evidence, find enough fossils to, for instance, in this case, locate and see key differences between the fossils of mammals, early mammals, when comparing them to the fossils of other tetrapods. Tetrapods, again, being those four-legged organisms. So things like reptiles, amphibians that are not mammals, there are specific skull differences. And so this is just a list of those four. The lower jaw is just one bone for mammals, where it's many bones in other tetrapods. And for some of you that are in my HBS class, you may remember we looked at the skull, and the skull is a bunch of different bones, but that mandible right here, that is that one bone. The lower and the upper jaws hinge between a different set of bones than those in other tetrapods. We as mammals have those three tiny, tiny bones the hammer, the anvil, and the stirrup, whereas the other tetrapods have just one. So that's a key tell if you just pluck up two skulls from the ground. And if we look at them, we could quickly tell if one is another tetrapod versus a mammal based on finding those three tiny little bones. And finally, we as mammals have differentiated teeth. We have our canine teeth, that pointy tooth. We have our incisors. We have our molars that all kind of serve different functions. When we speak about reptiles that have also those teeth in their skull, they are not differentiated. They're all mostly just the exact same shape as you make your way all the way around that jaw. All right, folks, I'm positive I've taken enough of your time. Other than what I'm giving you here, I'm not assigning anything other than prepping for Monday's quiz and just that. Just prepare, use the tools that you have. Gosh, talk to your classmates, do FaceTime meetings, have small group meetings. If you want to, Google Chat with me Friday afternoon. That's it, guys. Other than that, continue to take care of yourselves, take care of your families, stay safe, stay healthy. It sounds like more good news is starting to come out. Please take it in stride as we need to continue doing what we're doing. It is not going to be a, hey, there's good news, let's just go out and start seeing friends. That will lead to second spikes of numbers that we really don't want to see. So continue to be safe, guys. Thanks for everything. Miss y'all. And let's get out of here and enjoy the weekend. And my computer is, there we go.